You know, for decades, they were pushed to the very fringes of science, but now psychedelics are back in the lab in a huge way, and they're showing some absolutely incredible promise for treating our most stubborn mental health conditions. So how do these compounds actually work their magic? Well, let's dive into the cutting-edge science that suggests they might literally be remodeling the very architecture of our brain.
Okay, so first things first, a little context. The word psychedelic is fascinating all by itself. It was made up back in the 1950s by Humphrey Osmond, and it literally means mind manifesting. It was his way of describing this unique class of drugs that can so profoundly change our consciousness, how we see the world, and even our sense of self.
And that really is the billion-dollar question, isn't it? What is actually going on at a biological level when someone has one of these experiences? What does mind manifesting look like in the physical, messy hardware of our brain? That is the exact question we're going to unpack today.
Right now, we are living through what so many people are calling a psychedelic renaissance. And the reason for this explosion in research is pretty simple. These compounds have staggering potential to treat a whole list of conditions. We're talking depression, PTSD, addiction, even Alzheimer's, that have been incredibly difficult to manage for so long.
See, the core idea driving all this new research is that it's about so much more than just the subjective experience. I mean, the trip gets all the attention, right? But what scientists are discovering is that these drugs have profound and maybe even lasting effects on the physical structure and function of our neurons. We are talking about a literal physical change. To really get how that works, we need to talk about one of the most incredible features of our brain. It's neuroplasticity. This is your brain's superpower. It's this fundamental, almost magical ability of our neural networks to change, to adapt, and to reorganize themselves throughout our entire lives. An easy way to think about it is that your brain isn't some static, finished building. It's more like a city that's constantly under construction. It's always building new roads, tearing down old ones, and strengthening the connections that you use the most. That dynamic, ongoing process, that's neuroplasticity. And this is so groundbreaking that scientists actually had to coin a brand new term for it, psychoplastogen. This word describes any substance that can rapidly kickstart that kind of brain remodeling. And right now, the most powerful and well-studied psychoplastogens we know of are, you guessed it, classic psychedelics. So, what are these things actually doing on a microscopic level? How do they kick off this massive construction project inside our skulls? Well, animal studies have given us some incredibly detailed clues. Okay, let's really zoom in here. Imagine a single neuron in your brain. What happens in the moments after a classic psychedelic molecule, say psilocybin from magic mushrooms, shows up? Well, researchers have been able to map out a pretty clear and fascinating chain reaction.
It basically works like this. First, the psychedelic molecule docks onto a very specific spot on the neuron's surface, the serotonin 5H2A receptor. It's like a key fitting perfectly into a lock. This single action then triggers a cascade of signals inside the cell, lighting up two critical pathways known as TrkB and mTOR. You can think of these guys as the architects and construction foremen of the brain. They draw up the blueprints and give the order to start building new connections, sprouting new dendrites and synapses. And get this isn't just some fleeting, temporary effect. A 2021 study in mice found that just a single dose of psilocybin caused the rapid and persistent growth of new dendritic spines in the prefrontal cortex. I mean, these are the little branches that form connections. This suggests the brain remodeling could be long-lasting, which is, of course, a huge deal if you're hoping for lasting therapeutic effects. So we have this amazing, detailed picture from the microscopic world of animals. But here's where it gets tricky. Taking those precise findings from a mouse and trying to apply them to the incredible complexity of a human brain, well, that is a major scientific challenge. You really see the trade-offs in the research here. With animal models, we can get direct, cellular-level proof of what's happening. But the behaviors we measure, like the famous head twitch response in rodents, might not be a very good stand-in for a complex human experience. They might lack what scientists call face validity. On the other hand, with humans, we can study all those complex thoughts and feelings, but our tools for actually seeing the brain changes are much more indirect, much more zoomed out. And this one finding just blows the whole problem wide open. Scientists found that a drug called kedenzurin can completely stop that head-twitching behavior in mice who've been given psilocybin. But, and this is fascinating, it does not stop the psilocybin from actually growing new neural connections. This shows a massive disconnect between the behavior you can see in the animal and the underlying plasticity we're actually trying to study. Okay, so if we can't stick a microscope into a living person's brain, how in the world do we study these effects in humans? Well, we have to zoom out. We use a powerful toolkit of non-invasive technologies to get a big picture, macro-level view of the brain in action. And this toolkit gives us all these different pieces of the puzzle. We use MRI to look for large-scale structural changes. Functional MRI, or fMRI, lets us watch brain activity and communication networks change in real time. EEG and MEG let us listen in on the brain's electrical symphony. And we can even take blood samples to look for telltale markers of plasticity, like a protein called BDNF.
So this is really important. When we're talking about humans, the term plasticity takes on a much broader meaning. It's not just about growing new synapses anymore. It's about the end result, a measurable change in a person's behavior, their personality, or their way of thinking. This is how we connect the deep biology to the real-world therapeutic results we're all hoping for.
And this brings us to one of the most exciting ideas in the entire field, this idea of a window of enhanced plasticity. The theory goes that after a psychedelic experience, the brain is in this uniquely flexible, uniquely receptive state. And this could be a golden opportunity for psychotherapy to really work its magic, to help a person forge new, healthier thought patterns, essentially rebuilding their mental world on this freshly remodeled brain.
So we know psychedelics kickstart this neuroplasticity, and we have these tantalizing clues about how that links to real therapeutic change. But let's be clear, this is still very much a scientific frontier. There are some huge, fascinating questions that are driving the next wave of research.
The key questions researchers are wrestling with right now are things like, how do we bridge that gap between what we see in a mouse and what happens in a person? What is the exact link between growing one new synapse and feeling less depressed? And all of this leads to the big one, the most hotly debated question in the entire field, a question that could honestly change everything.
Is the profound, mystical, sometimes incredibly challenging subjective experience, the trip, is that essential for the healing to happen? Or could we develop a kind of non-hallucinogenic psychedelic that grows new neurons and provides all the therapeutic benefits, but without that mind-manifesting journey? The answer to that question could really shape the entire future of medicine.